reactivity case. That's going to lead to a discussion of two different types of compounds. Let's just actually draw them, and then I'll start talking about them. And let's draw these two types of compounds together. There's two carbons here and two carbons sandwiched between these things. These two compounds are very, very special compounds. We'll notice inside them that they have leaving groups, nucleophiles, leaving groups, nucleophiles, and these types of compounds can undergo very, very similar types of reactions we were just looking at. These are special and historically significant compounds. This is the basis for mustard gases, which were terrible, terrible chemical weapons first introduced to the world in World War I. We'll talk about how they react with biological molecules like the ones that we're made up of. This type of chemistry and understanding of these compounds ultimately led to this class of compounds that served as a basis for anti-cancer chemotherapeutics. So these particular compounds actually cause blisters, death, internal bleeding, cancer. And the understanding of how these reacted with biological molecules ultimately led to a series of compounds that could be used to react with malignant type molecules and things that are happening in our body selectively. These particular compounds, we'll start out with this one. It's a little bit simpler to discuss. It has very, very unusual reactivity with water. This compound has unusual reactivity with water. If we add water to this, either as a solvent, or let's think of it in terms of a nucleophile. Let's think of water as a nucleophile. This is our substrate. Chloride, at this point, you should realize, is kind of a poor leaving group. It's fairly electronegative. Iodide, bromide, tosylate, those things are better, but chloride's pretty poor. What about water as a nucleophile? The nucleophile trend requires we understand something about electronegativity. Chloride's kind of a poor leaving group. Water's kind of a poor nucleophile, but still somehow this compound reacts very rapidly to do this double substitution reaction. It's a little bit strange. Here's why it's strange. If we try to react these two things together, let's just take alkyl chloride with water. It'll react. It reacts quite slowly. And let's just set its relative rate here to 1.0. This is actually it'll work. It's actually quite slow compared to what happened above. And here is the comparison. It makes something clear to us that something's fishy about this compound where we have sulfur in it. Let's take this particular molecule where I've just put a sulfur in the fourth location away. So there's just an ethyl group between sulfur and chlorine. This particular compound also allows us to convert the alcohol, substitute alcohol in place of chlorine, and it happens 700 times faster. So this reaction is quite slow, but as soon as sulfur is put in here, this reaction accelerates 700 times. You're looking at this, is a little bit funny. How would sulfur have any sort of effect over here? It's not an electron effect donating or withdrawing effect here. What happens with these compounds and why they have such unusual reactivity is the following. These mustard gases were discovered to react internally with themselves. by SN2 reaction. So this is an intramolecular SN2 reaction. This is exactly like the reactions we were just doing with epoxides or with nitrogen attacking a carbon with a bromine group. Exactly the same thing. And what we make out of this is a very strange compound. It's like an epoxide. I guess it would be called like a thioepoxide. And this is kind of what we make. And this species here turns out to be very reactive. We have a three-membered ring. We have an atom with a plus charge on it. And if we add water to this, this ring will pop open very quickly. And the bad thing for the world, these compounds were released upon it, that both DNA and proteins are littered with good nucleophiles, like in the form of amines. Okay? So our biological molecules, such as DNA and proteins, have nucleophiles built into them, and usually the amine is the most powerful nucleophile in a biological molecule inherently. And so what happens to this is that this nucleophile, or water in the previous rate examples, attacks this, pushes the electrons back, and now we have either DNA or protein linked to this molecule. And this can happen again on the other side. And it gives rise to, just using DNA, it's a little bit easier to write here. Gives rise to another opportunity for the sulfur to form this highly reactive three-membered ring and a second molecule of, say, a living tissue or organism. Something from a biomolecule could react again. And what happens with this is that you end up with cross-linked DNA or proteins or other biomolecules. And this leads to cell death, blisters, tissue destruction, etc. So these types of compounds have this very, very special type of reactivity, and ends up, when you have cell death or cross-linking of molecules, DNA that shouldn't be cross-linked covalently, we get tumors, all sorts of other problems. We have this replication stops for DNA if it's covalently cross-linked. But this type of pretty wicked technology and wicked concepts and ideas were ultimately recognized that you could actually cross-link DNA, and under certain conditions and certain modifications, this could be used to therapeutic effect. And so other class of compounds emerged with this particular basic structure, and this can obviously undergo the same type of intramolecular SN2 reaction. And obviously, this too could react with some sort of biomolecule in the same fashion. Right? Could react in the same sort of fashion. But something is different about this nitrogen group that people discovered as they continue to work on this, is that this is less nucleophilic than sulfur. The reactions or reactivities are easier to control and manipulate. So it has lower reactivity. It's less nucleophilic than sulfur, and reactions are easier to control. slower, basically, is what it means. And ultimately, that this type of chemical compound could be used to great effect in tumor tissues to cross-link and stop tumors from replicating, right? So if you can cross-link DNA, it basically disrupts the replication mechanism. These general types of compounds, we're going to take a generic form here. And we're going to use R in a different context that we've used. We're going to use it very specifically, this R group. There's three major R groups that could be in place here. We're going to just draw these into our notes just for fun. We have a benzene ring here. Carboxylic acid, we're going to put this one in. Another one will have phosphorus. Have some really exotic functional groups today for our notes. Put this type of functional group in. And there's another one we'll put in here. It's a modification of the first one. It'll have a benzene ring. That's a little group that looks like an amino acid. And these compounds are actually, if we take this particular general compound and we replace this R group with this particular compound is good for chronic lymphocytic leukemia. It's actually a compound that's FDA approved to take. It's a very famous one. This particular modification is effective treatment for pancreatic cancer and ovarian cancers. So these types of molecules with a special reactivity that could be attenuated and tuned down a little bit could be controlled and put to the application on tumor tissues, treating tumor tissues in particular with these particular compounds to stop unregulated and uncontrolled cell growth, which is the hallmark of cancers. So this wraps up our discussion of SN2 chemistry.